Stem cell therapy following brain injury and neuronal loss is currently a hot topic in neuroscience research. In line with this theme, this video will be presenting the translational potential of a recent study conducted by Faulkner et al. At the Max Planck Institute of Neurobiology. Currently, neuronal cell loss due to injury or neurodegeneration cannot be repaired, and the ability of the adult brain to compensate for neuronal loss is limited. However, this recent study found the unexpected survival of embryonic neurons transplanted into the brains of adult mice. This raises hope for the possibility of using neuronal transplantation to treat diseases like Alzheimer's, epilepsy, and Parkinson's, along with brain injuries due to stroke. Transplantations of embryonic dopaminergic neurons have been promising in treating severe cases of Parkinson's, since these neurons have been shown to survive in patients. Some clinical effects in younger individuals have also been documented. However, the results are still variable at this stage. In terms of epilepsy, transplants of inhibitory neurons have been done within the hippocampus of adult mice with confirmed epilepsy. Results showed significantly decreased seizure occurrences and restored behavioral function. However, the purpose of this paper was to determine whether there is true functional replacement of lost neurons within the brain, and whether they are able to integrate into pre-existing circuits. This picture shows a single stem cell-derived neuron that has migrated away from its transplantation site in the cortex and has grown into a mature neuron. This study involved the transplantation of embryonic neurons from donor mice into the visual cortex of adult mice. First, the visual cortex of the adult mice was lesioned by injection of Lumiflor beads. The adult mice were anesthetized and unilateral craniotomy was performed. This injection was restricted to V1, resulting in selective ablation of this area. In order to be able to visualize the transplanted neurons, they were fluorescently labeled. In utero electroporation and viral labeling were used for this purpose. $25,000 to $80,000 embryonic cells were then injected into V1. Various methods were used for imaging, including standard optical imaging, two-photon imaging, monosynaptic tracing, as well as immunocytochemistry and immunohistochemistry for analyzing neuron morphology and histology, respectively. By 4 weeks post-transplantation, the majority of newly introduced neurons had acquired morphologies typical of mature pyramidal cells. They also saw long-term survival of grafted neurons and their stable synaptic integration. Importantly, they were able to establish proper incorporation of newly introduced neurons within pre-existing circuits. Efferent projections from the transplanted neurons closely matched the normal projectome. They also found quantitative similarities between the afferent projections of transplanted and endogenous neurons. In particular, there was strong innervation from the dorsolateral geniculate nucleus of the thalamus, which represents the most important connection for proper circuit function. Single cells of the visual cortex encode specific stimulus features, such as orientation and movement direction. Transplanted neurons were found to show selective responses to such stimulus attributes, which is highly indicative of their correct functional integration. This study has ultimately demonstrated the ability of transplanted neurons to integrate with great specificity into pre-existing circuits, sharing similar characteristics to normal development. Transplanted neurons don't require prolonged time for maturation or correct incorporation. These neurons were also able to receive inputs from brain areas known to project to the primary visual cortex and send out correct projections. Functional integration was also established as visual processing was observed. The transplanted neurons of visual cortex identity are specific, since no significant integration was observed when grafting these neurons into the motor cortex, indicating that successful transplantation required a specific regional match between the grafted and lesion neurons. This study ultimately shows that pluripotent stem cell-derived neurons can integrate into the pre-existing neural circuits. Therefore, damaged pathways have the potential to be reconstructed in the adult mammalian brain. Despite the fact that this was a very thoroughly carried out study, we will address some of the limitations and also how these results can translate into future research directions. First, it was noted that the entire study was performed on the V1 primary visual cortex, a region that is fully mapped out and well studied. This made it straightforward for the researchers to validate connectivity in the investigated neuronal synaptic pathways. Whether these promising results could be replicated in other brain regions remains to be studied, and we expect that upcoming research will explore this. Secondly, the article makes an important implication that should be elaborated upon. The transplanted neurons studied in this paper extended to their targets with great specificity, suggesting that guidance cues persist in the adult brain or are re-expressed after injury. This is a crucial finding since it implies that the natural guidance mechanisms that our brains express in early development might still be present or reappear when they are needed following injury in the central nervous system. If this can be seen through transplantations in other brain regions, this would mean that our existing knowledge of the brain environment following damage may need some reworking. Importantly, it was found that the tuning speed and the mechanism of neurons differs between the transplants and the endogenous neurons. They were found to be significantly slower than the endogenous neurons, which take a rapid 1 week to complete the tuning process. Infants achieve afferent neuronal wiring very soon after birth, with sharp tuning within 1 week, reaching adult-like values. Tuning refers to the process by which neurons selectively represent a type of sensory stimulus. This is how highly specific synaptic connections are formed between the retina and the visual cortex, for example. Feed-forward inputs that convey shared feature selectivity are initially coupled by gap junctions, but then replaced by the more conventional and slower activity-dependent chemical synapses. Transplanted neurons were observed to actually skip this step, meaning that their tuning mechanism was dominated by only activity-dependent remodeling involving chemical synapses. This could prove a key future research direction. In the case of vast cell death seen in stroke, for example, the mechanisms by which transplanted neurons incorporate into existing circuits could reveal new ways to treat patients whose cognitive functions would otherwise be lost following neuronal damage. In summary, this paper supported all of its conclusions rigorously with an enormous set of reference data and incorporated a broad spectrum of neuroscience, including neuroanatomy, neurobiology, and neural development. This makes it a fantastic paper for advanced neuroscience students as well as leading researchers to build upon with their own studies. It demonstrated entirely successful embryonic neuronal transplants into the visual cortex of adult mice with full integration into pre-existing circuits. Redoing these experiments in different brain regions and with slightly different transplants such as induced pluripotent stem cells could provide further improvements upon their methods. With further optimized research, these findings could open the door to viable treatment strategies for traumatic brain injuries, stroke, neurodegenerative disease such as Parkinson's, or even certain brain cancers in the future.